Hey guys, I know biology practical is a pain point for many students, so I'm going to make this video highly requested 7 must-know tips to acing your bio practicals. Students should always prioritize this type of questions in the practical paper. Do you want to know what it is? Ironically, it's the theory questions. I know it's practical, and that's like the main attraction of the paper. But examiners don't actually see what you did in the lab. They judge you based on what you wrote in the answer booklet. So it all comes down to what you write in the paper. Therefore, prioritize all the theory questions. They are way more important than doing the practical component perfectly. Want to know the #1 reason why students cannot finish practical papers? They want to do everything perfectly. They want to make sure every measurement is perfect. And when they make even one mistake, they like to redo that whole setup because they want to be super accurate. Trust me, I've been there. I know that you want to make it as good as possible, but unfortunately, it kills your grades. I have failed to finish many experiments because of this exact mistake. Let's call such a person Bob. So Bob's results are super accurate. He is so precise with all his measurements. He always checks measuring cylinder at eye level. He always pours perfectly every drop and he gets the perfect results. However, he doesn't have enough time to finish the paper, and as a result, he loses tons of marks. Now, compare this to Robert. Robert knows he won't finish the paper if he tries to be perfect. He measures things decently well, but not 100% perfect. As a result, he finishes his practical in 1/3 the time that Bob takes. And he spends way more time on the theory questions, making sure he spams every question with relevant keywords, and therefore he gets them done super well. Bob gets B4 for the paper and Robert gets A1. True or false? If you fake your results in a practical, you will lose a lot of marks. Actually, false. If you fake results and they are too far off, yes, you will lose a few marks for your lack of accuracy. But if you use those inaccurate results and do the rest of the paper with the correct methods, you will still score all those other marks. So if you have no time or you made a huge mistake in the practical and you need to redo, although it's not ideal, faking results is actually way better than starting from scratch because you would risk not finishing the paper. Hey, do you hate source of error questions? Don't worry, let me help you. First, we need to identify the source of error. This must be a problem that is built into the procedure's instructions that anyone doing will also face and suffer from. It's an inherent flaw in the experiment and it's not due to your own negligence. A good way to identify is to ask yourself, what variables should be kept constant but yet they are changing in this experiment? 3 main categories that the answer can come from. #1, inconsistencies of biospecimens. Example, you're using potato strips in the experiment, but it turns out these are from different potatoes. Then the contents of the cells would not be the same anymore and it's no longer a fair test. The second category is limitations of the instruments you are using. Example, they tell you to use a beaker to measure volume when in fact measuring cylinder is more accurate. Or they tell you to do Benedict's test and there's a color change, right? But color changes are subjective. We are measuring using our vision. Different people who look at it may call it a different color. So instead, we can use a color standard to compare the results to. The third category is influence of environmental conditions. Example is a photosynthesis experiment. They have a lamp shining light on the setups, which contain plants. They don't realize that the lamp is generating a lot of heat, and that is an environmental influence because temperature of the setups are being affected, and that would affect the rate of photosynthesis, and it's a significant problem. Remember, it must be an actual problem that's messing with the results. You won't get marks if your source of error is too trivial. When you start a practical paper, do you just follow the instructions exactly as you read them? Actually, the better way to do things is to read through all the instructions up till the very first question where you have to do something. This way you'll prevent yourself from getting confused and save a lot of time. The only exception to this is if you really have no idea what's going on. In that case, just follow step by step because the procedure is quite dummy proof and you will be able to produce the results they want you to get if you just follow the instructions. During your practicals, right, what do you do during the waiting time? Do you just stare at the setup and wait for the reactions to complete? If that's the case, you're wasting tons of valuable time and that's why you can't finish your paper. Use this waiting time to proceed with the next steps, or do a certain question, or prepare the next setup. This not only saves you time, but reduces your chance of making mistakes since you've carefully prepared the next step already. Never be caught doing nothing. If you haven't been doing this yet, this tip could save you from not finishing your next practical paper. Tell me, are you afraid of planning questions in practical papers? So long, right? It's 5 marks. Actually, planning is a commonly asked question, meaning it can be prepared for. Think of planning as like you are the setter now, and you are giving instructions for the procedure. Use this acronym ICROP. I stands for what is the independent variable and how you're going to vary it. Example, you would say set up an electrically heated water bath at 10 degrees Celsius, 20, 30, 40 and 50 degrees Celsius. So you've just stated that actually independent variable is temperature and this is how you're varying it. C stands for the constant variables. I know some students would like to just list a bunch of them near the start of their answer, but I prefer to weave it into my answer. This way I actually save some words because I don't need to restate them later. Example, I would say using a measuring cylinder, measure out 10 cm cube of solution X. This already implies a constant variable, that the volume of solution X is always 10 cm cube throughout my setups. R stands for repeats. So this is a very short point. You just say repeat steps blah 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 so that you get an average, and this increases reliability of results. O stands for what to do with the other 4 values of your independent variable. So I would say something like repeat steps what to what, but for the other temperatures of 20, 30, 40, 50 degrees. Finally, P stands for process the data to make a conclusion to actually answer the question. If they want you to find the optimum temperature for photosynthesis, then you will need to draw a best fit curve, then find the highest rate on that curve. So you must instruct the student to do this in your planning. You don't have to give the answer because you didn't actually do the experiment, but you must tell them what steps to take to get to this answer. A bonus tip that I have for planning is modify the original experiment as little as possible. Keep as much of the things the same. That way, you can play safe because you can be quite sure the methods they used in the practical question are probably quite sound. But if you change it too much and use your own methods, it may not be accepted. Want to know the biggest reason why students find practical hard? Because they're not familiar with such an experiment or the theory behind it. Just like when you come across a new math question type that has never been taught to you before, it's so hard and you take so long to do it. But for those question types which you've done many times and you're familiar with the method, once you see it, you're like, oh, is this question? Then you just do it so fast. The same thing applies for practicals. So go over all your past practicals you've done in school to familiarize. But I know many of your schools don't give you that much opportunity to practice in person because the school labs might be busy for other students. My secret to this is ghost practicals. This is where you take a new practical paper, read the instructions, and imagine doing the experiment. Imagine all the setups, the reaction, the color change, and so on. Then you will actually do that practice paper. So write the table, draw it out, record your made up results, plot the graph, and answer all the theory questions seriously. Even though you don't have lab access, but this would get you so much better prepared for practicals compared to everyone else. So those are 7 must-know tips to ace bio practicals. Do you have any special secrets to share with our followers here? If yes, please tell me in the comments. It will really benefit everyone else. All the best for your bio practical, and in the meantime, go check out this video here.